Okay, guys, so let's finish our lesson on DNA. We talked about the structure of DNA. Let's move on to DNA replication. Your objective is to be able to explain how replication occurs during the S phase of the cell cycle and how it allows the daughter cells. Remember, you get two genetically identical daughter cells from the cell cycle, and how they have an exact copy of the parental DNA. So when a cell is ready to divide, it must first copy its DNA. The process of making an identical copy of DNA is called DNA replication. This occurs during the S phase of the cell cycle. So again, if you fall down, you skin your knee, you want those skin cells to come back and be the exact same type of skin that you had prior to falling down, right? This occurs during the S phase of the cell cycle. And recall the S phase is a subphase of interphase. You have G1S and G2. S phase is when DNA is replicated. It occurs in the nucleus of the cell, and this DNA replication ensures that each new cell will have the exact same DNA as the original cell. Every cell in your body has the exact same DNA, and it must be replicated first before it can make a new cell. So the first step in DNA replication is to unzip the DNA molecule. The hydrogen bonds, remember, they're weak bonds. This is why. They're weak because they can be unzipped or opened. The enzymes help free-floating nucleotides to fill in the missing base pairs. They find complementary bases and they bind them according to the base pairing rules. DNA polymerase, that's a term that you'll be responsible for. The enzyme DNA polymerase, they help to find that complementary base according to the base pairing rules. Of course, I'm simplifying it, but you do need to know whatever we discuss in this video. And the third step is synthesis. Two identical DNA molecules are formed in the nucleus. Each will have an old strand and a new strand. We call this the semi-conservative model of replication. It was an idea that was presented by Watson and Crick. It means that the two strands of the parental DNA molecule separate, and each strand acts as a template for a brand new complementary strand. The new DNA strand, this is the new DNA strand, it consists of one original strand, or the parental strand, and one new strand. And remember that the result of the cell cycle of mitosis is two 100% genetically identical cells. This is how we get the same exact DNA. We use a template and a new strand for that new cell. So I want you to copy this picture in your notebook. Pause this video if you need to. And again, DNA model has been unwound here. We unzipped it. DNA polymerase attaches the new bases, complementary base pairing, to each template on the old parental strand, and then you have your result. Two 100% genetically identical daughter cells. Again, the DNA unwinds and unzips. That chemical bond, what's that chemical bond called? It's called hydrogen, is broken. The two strands are the templates for the new nitrogenous bases to attach, right? They are templates. We call that the semi-conservative model of replication. And once the new bases join, two identical DNA strands are complete. One with the old strand, the parental strand, and one the new strand.